Okay, so in this lab demonstration, I want to go over two tests that we would look at, things we look at in a soil test. We've already discussed cation exchange, so CEC, but what I want to talk about is base saturation as well. So I'm going to introduce you to that concept. Um, so this picture down here will sort of be reminiscent of what we talked about with the cation exchange, the idea that positive ions are attracted to negative charges on the soil surface. Um, what base saturation does is it basically looks at what is currently stuck on your soil. So if you send in this soil clump that's in this picture, let's say you just bought that clump and you put it in a bag and you send it off to the lab, they're going to expose that clump to an acid, and they're going to knock off whatever is there, right? So things like calcium, potassium, and they're all going to end up in a beaker, right? All of them floating around in there, and then they're going to use probes and tests to measure each individual element, and they'll find out how much magnesium is in there, how much calcium, how much potassium, etc. And that gives them a snapshot of what is actually on your soil, not in it in terms of the water solution that's around it, but what is on your soil. That's the base saturation. So if there's 24 negative charges and half of those charges are occupied by a calcium, then it's 25 or 50% calcium base saturation. Okay. On the non-audio link, this video link will work to take you to something that will help you understand that. In the extra content folder, I've got two videos by Ag PhD, a couple of kind of goofy academic farmer types, but they do really good videos and they do a very good explanation of what they're talking about when it comes to cation exchange capacity and base saturation. So there are some target ranges that are ideal. So for calcium, you're looking at quite a bit occupying the spaces in your soil on your charged surfaces. So 65 to 80% is calcium. And then when you're looking at magnesium, 12 to 25% magnesium on your base saturation. And then potassium, and then 10% or less with hydrogen. Why would we want hydrogen? Hydrogen's an acid, and by having some of the charges on your surface being occupied by protons means your soil will buffer the pH a little bit. It's got some reserve in it, which is a good thing. If it was 100% topped up, you wouldn't be able to add anything to the soil. It would just leach away. So we want to make sure there's some available spots. If you do watch the videos, which I hope you do, with Ag PhD, they'll get into this, but I'll just briefly explain to you one thing. Calcium, magnesium are major nutrients when it comes to soil charged surfaces. And they have, not only are they a nutrient, but they have physical effects on the soil. Calcium is a very large atom, so think of it as a giant beach ball, and magnesium is a very small atom, like a little silt grain. Very different size. I know often we don't really think about atoms being different sizes, but they are. And when there's lots of calcium in soil, those large atoms actually open up the soil and improve its porosity. So water drains through it better. It gets better aeration. When you have lots of magnesium in soil, because it's small, it helps pull the particles closer together. Making the soil waterlogged and less prone to drainage, and then you end up with less oxygen. So an interesting trick that farmers have learned over the years is that by adding to soil that's not draining well, by adding a good dose of calcium to the soil, it fixes a nutrient problem that's out of balance, and it loosens up the soil. And to give you a personal example, a relative of mine drills water wells for a living, and other kinds of wells, sampling wells and stuff. But and oftentimes when you're drilling, you come to like a heavy clay section, and the bit doesn't push through that easily, and it gets plugged up, and it's a lot of work to get through that layer to keep drilling deeper for water. And what they do in the drilling industry is just like what we just talked about. They pull the bit back and they dump calcium chloride down the hole, and the calcium reacts with the clay particles, opens them up, and the knife or the drill will actually drill through the section like butter within a matter of minutes. That's how quick the reaction is. So calcium opens up soils, makes them more porous. Magnesium makes them really tight. So if you get a base saturation analysis and the data comes back and shows that you have more than 25% magnesium in your soil, you may have a tight soil that's not going to drain very well, and you're likely not to have enough calcium for your plants. So this slide's obviously very small, so this is a soil analysis that we did on college rhododendrons at different areas. And you'll notice down here it's clay loam, sandy clay loam, loam, and loam. So these are different sections of the college campus. If we zoom in to over here, you'll see on this column it's cation exchange capacity, CEC, right? So that's your total capacity. And then over here we have our base saturation or cation saturation. And what's interesting is looking at these numbers. We've got 1.5, 0.4, 1.2, 0.6% of potassium, where potassium is supposed to be in the 4 to 8. So I'll see if I can write this, 4 to 8%. So potassium's a little bit low. Magnesium, on the other hand, is fairly high. We have some two soil samples that are one's at 29.5, and the other one's all the way up to 37, where usually it's no more than 25% magnesium, right? So we have a problem there. Calcium, looking at calcium at 25.9, 23.3, 31.9. What is calcium normally? We want normally 65 to 80. I'm getting better at writing with my mouse, by the way. I don't know if you noticed that. 65 to 80% calcium. So we're way low in the calcium here. So these plants are going to be having some problems. And then hydrogen. Remember, that's the buffering capacity, the ability of the soil to store more nutrients. It's like free parking spots with little pylons holding that spot. They're all the way up at 50, 54%. There's a lot of unused space in that soil. It needs some nutrients. And what does it need? It needs a little bit of potassium, but it definitely needs calcium. So one of the simplest things you can do in this context is add lime. So what does lime do? Okay, so lime is calcium carbonate. That's this molecule here, calcium and a carbonate molecule. They react with hydrogen. Remember, those are those H's on your soil, and they react and form different things. The calcium is released, CO2 bubbles out into the water. This is basically like adding baking soda to your cake. When you add it, the CO2 is formed from instead of calcium, it's sodium carbonate or bicarbonate, and the CO2 forms bubbles, which makes your cake rise. And so in soil, when you add lime, calcium is released. It binds to the sites that were occupied by the hydrogens, and the hydrogens are turned into water because they react with oxygen, and the rest of the carbonate is turned into carbon dioxide. So there's a chemical reaction that happens in the soil, and the benefit, which you have to remember, you don't have to remember the chemistry per se, but the major benefit is that by adding lime, we add calcium to the soil, we consume some of the acidity, right? So we're affecting pH, and we are impacting base saturation. So here again, how is removing hydrogen from soil colloids helpful? Remember I said they're kind of like a parking spot, like a pylon, right? So this is sitting here, not doing anything. If I add lime, boom, I consume the protons and I replace them with calcium. But if we remove all of them, then our soil has not got a lot of buffering capacity. But it doesn't have to be that thought through. But basically, adding lime is a very useful thing, especially to soils that are not draining very well and are showing calcium deficiencies. So we raise the pH of the soil because we have fewer protons present. Right, so if we're dealing with an acidic soil, so there's that. We add calcium, we talked about that. We increase the availability of charge sites by consuming protons, allowing soils to better buffer pH. So for example, sandy soils which have very poor cation exchange and base saturation, their pH changes rapidly. And farmers really struggle to keep the root zone at a happy pH. Where heavy organic soils tend to buffer their pH and they're much more stable because they have far more cation exchange capacity. So if you have a sandier soil, this is a much more important exercise for you. All right. There's a couple of scenarios that are coming up. I'm going to do separate videos and insert them all between these slides. And that will help me do what you would have done in the lab, which is sort of demonstrate them with little simple models. But hopefully this is helpful. Okay, so I've got two sponges here with some wooden kebab sticks in them. And I'm going to sound repetitive. Some students are going to get this right away and some are going to be confused. So I want to go through this over and over again to make sure you all get it. Essentially, each of these sponges represents a soil colloid. It could be a piece of clay, or it could be a piece of organic matter, a chunk of compost. This soil demonstrates a soil particle that has very poor cation exchange. Each of these spikes represents a potential site where a cation could bind. So this is a negative charge, negative charge, negative charge. So calcium could bind here, here, or here. Or potassium could go here, here, and here. Whereas this soil, which represents a high cation exchange capacity soil, has lots of negative charges on the surface. And it can bind lots of potassium, calcium, magnesium. Okay, so this is an example of a high CEC soil and a low CEC soil. Okay, so in another lab scenario, I'm supposed to demonstrate what it would look like if you were taking a low CEC soil, like this sponge, so it's got very poor cation exchange capacity, and we want to increase it. And so using the exact same sponge, I'm looking lazy here, but really it works, we can add humus. So by adding compost into the soil, boom, now we have our regular soil particles and what we've added. And look at that, we're happy. We've got lots of cation exchange capacity. So by adding compost or biochar, we have improved the cation exchange capacity of this soil. Okay, so before, after. Okay, the next lab scenario says we're supposed to demonstrate what a soil would look like if it had 50% potassium, 50% calcium base saturation. So what I've got here is I've got 1, 2, 3, 4. Oops. And 1, 2, 3, 4. Let's say the yellow is calcium and the green is potassium. We have 50% of the available charges, that's each stick represents a charge. 50% are calcium and 50% are potassium. I don't know if I got those colors reversed, but I think you get the idea. Okay, so if we take this soil sample to a lab, they're going to wash it with acid, which is going to knock everything off the soil into a beaker, right? Then they're going to go into the beaker and they're going to count out how many of each was in the soil. And they'll say there's 4 of the potassium, 4 of the calcium. Our base saturation is 50% potassium, 50% calcium. Okay? So that's how base saturation works. That's kind of what it looks like. Okay, in this other lab scenario, we're supposed to demonstrate what happens when you treat a soil with lime, in this case calcitic lime, so calcium carbonate. In my sample soil here, I've got a calcium, potassium, 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 and I have 1, 2, 3, 4 sites that are just negative charges with hydrogens attracted. We can't see them, I don't have a color. Let's say there's 1, 2, 3, 4 hydrogens that are binding those negative sites.
You won't ever have a negative site by itself. It's always going to have something attracted to it, and it's always going to be a hydrogen if you don't have any of the cations around. So along comes our calcium carbonate in the water, right? It reacts with each hydrogen. So let's say it reacts with this one. It's going to take it off, and it's going to replace it with the calcium. And this hydrogen is going to turn into carbon dioxide and water and be gone. And then another calcium carbonate is going to come along, and it's going to remove that hydrogen and replace it with the calcium, and it's going to release water and carbon dioxide. Another one's going to come along. It's going to take up this hydrogen, right? And it's going to release water and carbon dioxide. And another calcium carbonate is going to come along, and it's going to take this hydrogen, and it's going to leave water molecule and carbon dioxide are going to disappear. And you're left with soil that has much more calcium attached to the negative charge surfaces and far fewer protons. In this case, I removed all of them. Okay, so that's what happens when we add lime. Okay, so I've devised a little experiment to demonstrate cation exchange capacity. And what we've done here is this is standard peat moss, your high porosity mix with your bit of perlite in it, around 20% perlite. And I've taken that and I've mixed it 50% with vermiculite. This is a bag of horticultural vermiculite bought at a garden center. If you look at vermiculite under a microscope, it looks a lot like clay. There's lots of thin layered sheets of mica. Vermiculite is usually added to soil to hold water. So it's put over top seedlings to keep them from drying out at the surface. You don't have to irrigate them as much, which disturbs the soil. It's used for seedling mix, again, to have consistent moisture levels. It's also used in soil mixes where you are worried about water loss, so crops that are really sensitive. It's expensive, so you don't often see it used. You don't often see it used in broader production, like it's a plant that needs to be kept wet. There's other ways using detergents and stuff, which we'll talk about another time. But vermiculite is available in high value things like seeds and plugs. It is used. But one of the properties of vermiculite is because there's these thin sheets of mica is that they have lots of negative charges. So I have over here, let me turn my meter on. This is, I'm going to do a pour through analysis here. This is my fertilizer solution that is sitting at an EC of 4.5. So let's set this aside and let's water in the standard porosity mix. I've given it, I'm going to go halfway. Okay. And then I'm going to go halfway with this. Not quite. All right, I did a little bit more in the vermiculite one. All right, so we're going to let them do a pour through. And we'll see what comes out the other side. If the vermiculite has enhanced CEC, less salt's going to come out the other side of the pot, right? Because the negative charges are going to hold on to all the cations that are in the fertilizer solution. So let's take this off. Let's pour some water through. Okay. To the next one. There's a little coming out for my excess, but that's okay. We're going to do a pour through on what the soil's holding. I don't really want necessarily what the volume's holding. So let's wake this up. Take the sample off and measure the EC. What came out the other side? And lo and behold, what came out the other side was 4.5. Exactly what we put in. Okay? I'm going to dump this into here and maybe to try to be a little bit empirical, we'll give this a bit of a rinse. Dump it there. I'm going to give my probe a bit of a rinse. Okay, let's put this here, and this really could be a lot more empirical, but you get the gist. Okay, let's get our probe in here. And the EC 3, an EC of 3. If you don't believe me, I don't know if you can see it on there, but I'm not lying. An EC of 3. So what's happened is the vermiculite has held on to those cations in the fertilizer solution. Now the anions will continue right on through, so you're going to get some higher salt levels. But what we've done here is we've improved the cation exchange capacity of peat moss by adding vermiculite. Hope you found that helpful.